thank you very much for the opportunity to interview you. As I said, this is the third in a series of interviews we've conducted in recent weeks. Liza Royden, breast surgeon and breast cancer survivor. Then last week we interviewed Rory Catlin-Jones, science correspondent of the BBC and Parkinson's sufferer. And now we're into the heart of science and clinical practice with your good self. And obviously, I know you from Twitter, and I know you're very active on Twitter. In fact, actually, we even interacted this afternoon. It's sort of a good segue to the first question, which is for people obviously who don't know you, maybe you could give us a little bit about your background, which was also a topic that we exchanged on this afternoon. Particularly how you got into research and clinical practice. I think I understand about 5% of what's on your CV. So maybe you can explain it and what motivates you in the fields that you work in, because you work in many areas.
Well, yeah, thanks very much for the invitation. It's great to have a chat with you, because we have chatted several times, but only via Twitter. So, yeah, my name's Stefan Marciniak. I'm Professor of Respiratory Science at the University of Cambridge. I'm also a respiratory consultant, that's a British for an attending, in respiratory medicine at Addenbrooke's Hospital in Cambridge and Royal Papworth Hospital. So what's my background? My background is it was relevant for a discussion on Twitter today, because somebody had said that people from working class backgrounds shouldn't go to Oxford. I was from a working class family, first person in my family to go to university. I happened to have the good fortune of being very bad at sport, so I had nothing to do except study. So I passed lots of exams and went to university. I really enjoyed the science as much, if not more, than the medicine. So when in the very early 90s, Cambridge was setting up a combined MB PhD, that means where you could do medical qualifications and research at the same time. It's a very American model, but it was new to the UK back 30 years ago. I was one of the first people in that. So I ended up graduating after 9 years rather than 6 years with a PhD and a medical qualification rather than just a medical qualification. I worked in a variety of specialties. I met some very inspirational respiratory doctors along the way. So I became a respiratory doctor. And I never fell out of love with science. So I kept popping back to the lab, and I did some research in the UK. I did a few years of research in the US. I then set up a lab in Cambridge well over 10 years ago, and we study some basic cell biology. But I also, in my medical practice, I probably do more research on the clinical questions than I would have done if I weren't also a scientist.
Okay. And I mean, for me, when I looked into your background, and there were links, you've got a lab, you've got a clinical practice, you're a researcher. It's very broad, because I mean, and you're doing protein folding as well as doing clinical research, as well as doing clinical practice, and also you're a fellow at St. Catherine's as well. So. I mean, first of all, it's incredible how you manage to do so much, as well as get onto Twitter every now and again and do interviews with us. Can I maybe? We've obviously got people without scientific backgrounds who will be listening to this video, watching this video. What's the sort of difference between the researcher and the clinical practice?
Oh, that's a really good question, and I could get myself into hot water. I think that research, I love the research because it's very creative. It lets my creative side out. If I could paint or do music, I'd probably do that. But research is more creative because you have to imagine lots of things that nobody knows, design experiments, and most of the time it turns out you're wrong. So you have to keep coming up with new ideas. And you're literally making it up as you go along. And that's research, but it's really, really fun because it's discovering new things. Medicine is, you don't know what's wrong with the person at the beginning, and it takes a while, but it's not often that you come across things that people haven't discovered in the past. Very occasionally you do, and that's one of the points. That's why it's quite good to be a researcher and a clinician, because you can see these opportunities and then turn clinical practice into a research opportunity. But in medicine, you're applying known knowledge to figure out very difficult questions and then fix them. Whereas in research, you're trying to create new things, and that's more creative, I think.
Yeah. So sorry, Laura, go ahead.
Sorry, you sort of touched on it there, but I wondered if you could expand on it a little bit. The benefits of having that clinical interaction to the development of your research work. Do you think that that's one of the limitations, perhaps, of how sometimes research is carried out, perhaps more in a silo, in a slightly detached way? What's your sort of feeling about the interaction between the two?
So research in general, I mean, the best research is done when it's really interdisciplinary anyway. So medics and basic biologists, or basic biologists and engineers. And my research lab, when I've got my research hat on and we're doing basic cell biology, we have lots of collaboration with physicists and engineers. We have research grants with engineers to try and use nanotechnology to develop new treatments, and we have collaborations with physicists to develop new ways to do microscopy that hadn't been done before. With medicine, I think medicine is a fantastic biological training. You learn an awful lot about many different things. You learn physiology and biochemistry and anatomy, whereas people who do basic science tend to learn one thing very, very well. So I did like the medicine because I got a very broad biological training. And I like the science because I can drill down on some small thing and become an expert in a small area. So I run the MB PhD program now, the one that I was in 30 years ago. I run it in Cambridge, and when I'm talking to, sorry, my dog's wandering around making noise. When I'm talking to the young kids thinking of applying, I say that it really, even if you end up doing pure medicine, if you've done research in the past, I think you think slightly differently. Maybe a pure medic would disagree with me quite reasonably, and that is that when I see a patient with something that we can't get to the bottom of, I see that as an opportunity to do some research and figure what's wrong with them. There was one patient I'm thinking in particular. She had an inherited pneumothorax. That's a popped lung. That's what some of my clinical work is about. She didn't really fit what the textbook said. And so we were able to do some research. We were able to look at the genes that were potentially involved. When we found some abnormalities, we were able to look at those in the lab, prove that they were defective, and then go back to her with a new diagnosis. And I think that's something that I would have struggled, I could not have done if I didn't have a lab, and I probably wouldn't have thought of doing if I hadn't done research training.
So that's a sort of interesting, because I thought that was really interesting about your profile, because most of the time, most people in clinical practice will just be doing that. So as you said, it's sort of like using the known in an unknown situation, whereas as a researcher, you're constantly questioning everything almost. Yeah, yeah. This is a constant place of total uncertainty in many respects. Yeah. And the area that you're researching in, you mentioned, I'm going to mispronounce this, pneumothorax.
Yeah.
And this is you said it's a popped lung. Can you?
Yeah,
I was going to say expand on that, which is probably
expand. Yeah, well, that's if you fix it right, it does expand. So that's my clinical side. So and I do research in it simply because I can't help myself. But my clinical, I'm supposed to be. I joke that I'm 50% clinical research. I run the pneumothorax, so I'm 50% lab, and I do 50% teaching, which shows I do a lot of the teaching in the evening. But when I set up many years ago, when I set up my clinical research, I was being paid for by research fellowships. So unlike people who are employed by the NHS, most researchers have to pay for themselves with their grant income. But it did have the advantage that I could spend a small amount of time doing medicine and a lot of time in the lab. So I had to do something small and niche. And one thing that I found very, very interesting as a trainee respiratory doctor was the inheritance of this condition where the lung pops spontaneously.
I think you described it, yeah. Mm.
Yeah, so the lung becomes, particularly in tall, thin young men, not exclusively, but usually in tall, thin young men, they can spontaneously have one of their two lungs pop and deflate. And it can be quite painful. It's unpleasant, but it's more frightening than dangerous usually.
I assume. One hopes.
Yeah. So the young people who have got the other lung is fine tend almost always do extremely well. The people who suffer from pneumothoraces are people who've smoked all their life, and they get them later in life for different reasons. But the interesting thing, the reason that my clinical practice concentrated on this is that lots of patients, when I was training, when they would come in, most of my patients come in with their mothers, even if they're 25, 30-year-old lads. They usually come in with their mum for some reason, like pneumothorax patients. They're lovely. And as they were leaving, often the mum would say, you know, your Uncle Eric had one of those as well. And that was very interesting. And the textbooks say that there's one condition that causes this, and it's a genetic condition called Marfan syndrome. But almost all of them did not have Marfan syndrome. So it was interesting. And most people have left that. They just said, fine, that's interesting. But when I set up the pneumothorax service in Cambridge, I then started looking into more detail in this and working very closely with colleagues in clinical genetics, and then using access that I have in my lab to more advanced diagnostics. Then the 100,000 Genomes Project was created, and the 100,000 Genomes Project does exactly what it says on the tin. The NHS wanted to sequence 100,000 genomes, and partly to look at cause of cancer, but also to look at rare diseases. And after a while, I managed to convince them that the pneumothorax was a great opportunity, because we had lots of patients that I'd thoroughly investigated. I could show that they did not have known genetic disorders that are in the textbooks, but they had an obvious genetic cause for it, and we could start seeing some. And we're working. We now have been analysing many of those with the 100,000 Genomes Project, and we're making some advances in that area. And then after a while, talking about it often at conferences means that when people have one of those patients in their clinic anywhere in the UK, and sometimes I think my furthest referral was from New Zealand, then people get to know that you're interested. They emailed me with these patients, sometimes the patients, but usually their doctors. And then my clinic then starts filling up with these interesting people that we can try and make a diagnosis on.
Okay, and you direct, I think you call it the Rare Disease Collaborative Network for this particular topic.
So I've run this pneumothorax clinic, which has been becoming increasingly a place where people would refer patients with these inherited hot lungs pneumothoraces. And then last year, NHS England, which has about 8 of these rare disease collaborative networks, which allows hospitals that specialise in rare diseases to collaborate across the UK. So although it's NHS England, my collaborator is a chap called Kevin Blythe. He's a professor in Glasgow. And so we set up the RDCN, the Rare Disease Collaborative Network, in familial pneumothorax. And as I said, there are about 8 others that cover different diseases. But what it means is that every month we meet virtually, so a bunch of doctors from Cambridge, a bunch of doctors from Glasgow via a secure web link. We discuss the difficult cases that we've had in our clinics in the last month. But increasingly, doctors in other hospitals around the country, perhaps much smaller hospitals that don't necessarily have the resources we do, or the geneticists with the experience we do, will dial in and present their unusual case. And we're finding that we're able to increase the likelihood of making diagnoses. So that's really useful from a clinical point of view, and improving equity of access to specialist care. There's no reason why somebody out in the middle of nowhere needs to be referred either to Glasgow or to Cambridge to see one of us. They can see their local doctor, and then they can pick our brains at this meeting. But in addition, and over time, we're going to accumulate more and more people who don't fall into any of the existing disorders. And that's another benefit of these so-called RDCNs, is that by grouping together patients who have these genetic disorders that we haven't yet figured out, makes it more likely that we'll be able to identify new diseases.
I think you've picked up there so many of the elements that we're really trying to capture at Care Circle. It's interesting because I had a question about the network as well, and I know that the NHS is increasingly moving towards this format of trying to have this collaboration amongst specialties. And you've mentioned sort of a range already in this interview of scientists and clinicians trying to work together. So I just wondered, to what extent you think that they're helpful, and what the potential is to have more of these networks to serve more of these conditions that perhaps haven't been identified as having a genetic component, or perhaps aren't being sort of adequately catered for just with the format of the NHS.
Yeah, so,
because of their rarity.
Yeah, so I mean, I'm one of the biggest defenders of the NHS. I think it's one of the greatest ideas anybody's ever had, and I'll defend it to the hilt. It's very, very, very good at looking after a whole variety of disorders, but increasingly, because of the success of healthcare, there are many more people being found to have rare disorders. If you go, this is an interesting statistic that a geneticist friend told me, is that if you go to the neonatal intensive care, a large number of the children on the neonatal intensive care will be there with an extremely rare disorder, because they wouldn't be there if they had an ordinary condition, because most of them will be fixed. And so increasingly, very rapid whole genome sequencing is being used, certainly in Cambridge, to make diagnoses of unfortunate kids on the neonatal intensive care unit. In most patients in any respiratory doctor will see any respiratory doctor in any NHS hospital and get fantastic care. But if you look across the whole UK, there'll be a large number of people with rare diseases. And it is inefficient and unrealistic that every single respiratory doctor is going to know about the incredibly rare disorders that I look after, for example, just about pneumothoraces, in the same way that I couldn't look after the incredibly rare disorders that affect the immune system or many other things. So concentrating a lot of expertise in a small number of people is good, it's efficient, but then making it accessible to everybody else needs to be done. And that's the idea of these NHS England Rare Disease Collaborative Networks, or RDCNs. And I said there are 8 now, and the plan of the rare disease, I'm getting an echo, the rare disease, I think it's called the highly specialised commissioning within the NHS, you'd have to look that up, I think, because I've forgotten the name, is then trying to increase the number of these year on year. So there's one, for example, for cystic lung disease, run by a colleague of mine called Simon Johnson in Nottingham. And there's another one for abnormal blood vessels in the lung called HHT, run by, I think, Claire Shoflin in London. So these, but because they can all be done virtually now, lots of doctors, they don't necessarily need to refer them to me. They can just email us, be put on the list of patients we're going to discuss that week, and anywhere around the country dial into them.
And you mentioned obviously rareness of disease. What about rareness of case when you may get a comorbidity? Do you have cases where somebody presents with a case, but there's something else going on as well? And then how do you deal with those cases?
Yeah, it's very often that complicated things, but the easiest way to is having seen lots of people with lots of and getting lots of experience. So when I see people with, because I run a pneumothorax patient clinic, and I see dozens of patients with pneumothorax every month, whereas other respiratory doctors will see a few, several every year. So it's not incredibly rare, but I will see dozens every week. I can then see patterns for me. So I can see, well, this particular unusual feature is happening more often than chance. So maybe there is a new condition, maybe this person has mild learning disabilities, maybe they have a particular cardiac defect. It was something along those lines that brought pneumothorax and Twitter and research together in my life a couple of years ago, when a couple of registrars, that sort of trainee specialist doctors, contacted me during the pandemic, during the first wave of the pandemic, saying we've seen, each one independently had seen a patient with COVID, so COVID, and pneumothorax, and they said, is this a thing? And I said, well, pneumothoraces can happen during chest infections. It's actually very uncommon. After viral infections, it's also very uncommon. So the fact that 2 different registrars during one pandemic wave had contacted me, I thought was odd. So being a thorough Twitter addict, I just asked without making any confidences, I just called out, and there's a tweet out there from sort of, I don't know, March or April, no, probably April of 2020, where I said, are people seeing pneumothoraces in COVID? And if so, DM me. And then there was this wave mainly across the UK, but also in other parts of Europe, of people saying, yeah, we've seen them. So we were able to aggregate all these people with lots of individual cases of pneumothorax and COVID. We did some research based on that. We estimated how prevalent it was, And then it was only the next year when I looked across the data from 130,000 patients in the whole of the UK that we were able to find that the Twitter study that we initially did was correct. So that's an example of, by getting lots of people together and getting lots of experience, you can see when new links are true and when they're just chance.
Yeah, you've just described one of the use cases of Care Circle, because we've started building tools to do that. So it's interesting. Yeah, exactly. And then I think it's an example of how researchers can work with patients directly to get sort of almost live information. Just to continue just for a second, because I know you do a lot of things, which we'll come to in a second, but on the pneumothorax, there's a pneumothorax day apparently at the 30th of what's that involved? How are you involved?
So when we launched, it was a little bit of fun, but it was actually has some sense to it. When we launched the respiratory, the pneumothorax, familial pneumothorax rare disease collaborative network last year, we just decided this was it. We're going to do some bit of fun on social media, and so we decided to create what we grandiosely called World Pneumothorax Day. So it was going to be a few people tweeting. Some charities got interested as well. So there's a charity that funds research in rare diseases called the Myelofibrosis Trust, and they're associated with the Bert Hogder Bay Foundation. Bert Hogder Bay is a rare genetic disorder that causes pneumothorax, and so I see a lot of patients with this condition. So they also got very interested. So they thought this was a great opportunity to make doctors think about a rare disorder, familial pneumothorax and Bert Hogder Bay syndrome, and make it slightly fun and people to participate. So they created lots of beautiful web art and things, and they had interviews with their patients and members of their with Bert Hogder Bay Society on their website, and they started tweeting about it. They told me that one day last year was when they got the most hits on their website in the entire year. So, and then as we were tweeting about it, think pneumothorax, think genetics, think this, that, the other, lots of doctors around the country, and in fact around the world, started joining in. And so, friends over in Oxford who didn't know about this day, they started talking about it. And the people, so during the course of the day, this thing that we created, World Pneumothorax Day, turned into a thing. So we're doing it again this year. I've already had a couple of short videos with the Bert Hogder Bay Foundation. I know the Asthma Lung UK are also going to be participating, and so it's hopefully going to be an annual thing, and it's an excuse for doctors and charities and patients to sort of talk to each other, but also make people aware of rarer conditions that they might not have heard of otherwise.
It's fascinating actually hearing you talk about bringing together all those elements of the ecosystem, because I think that's whenever you tackle anything in healthcare, it is recognizing all of those stakeholders and the role that they play. I was watching something last night from the film archive, actually, of when the NHS was first conceived and the debate that happened around that with doctors. One of the concerns that was raised by doctors at the time was that the NHS might actually break down the relationship between patient and doctor. And I think, like you, we're huge defenders of the NHS and health equity and everything that the NHS does to support that. So clearly we've seen that hasn't been the case. But I have been reflecting, listening to you speak about all of the technologies that are in place to really support modern medicine and all of the different ways that you're employing them. But I wondered what your thoughts were on sort of some of the negatives around technology, and particularly the way that perhaps social media and online media have managed some of the information sharing, the misinformation. We saw it particularly with COVID, and we still have the limitations that social media is exacting on us as a population in terms of what we can share and what we can learn about the pandemic. And obviously that particularly affects your specialty. It's
a challenge. So the social media gives everybody a soapbox. So people who know what they are talking about, experts, can stand up on this soapbox and tell people about it. And patients who've got their lived experience of a disease can also talk about it, and that's also incredibly valuable. But also people who have no idea what they're talking about can do the same thing. And it is very difficult when you're dealing with small little tweets or somebody who just amassed a load of followers, and on social media, often followers can be mistaken for gravitas, and so people who've no idea what they're talking about can spout, and people will believe them. Worse than that, and more sinister, is some people have a different agenda, and they are trying to sell you something. They're trying to sell you their book. They're trying to sell you their version of snake oil, their whatever. It's often a diet, for example, and they can then be quite active on social media, and it's difficult sometimes to counteract them because they can amass a very loyal following. It's very easy, and I'm going to get into a lot of trouble here, but it's very easy for people who believe in conspiracy theories to start feeling that they have some secret knowledge and that they're superior to everybody else because they know something that all the doctors don't know, and all the patients don't know, and all the governments don't know. And of course they don't. It's not true. But it's almost impossible to convince them on somewhere like on an echo chamber like Twitter that they're wrong. So yeah, so often these people will say something completely outrageous, either because they're trying to sell something or because they're following somebody who's trying to sell something. It's difficult for me not to challenge them and point out the truth, but that tends to degenerate very quickly into quite a difficult argument, and there's no point. It gets quite unpleasant. So very often after a few difficult interactions, I will just mute the person. So I think that is a problem. I think so the spread of misinformation,
yeah,
and the spread of ignorance.
Well, this was a topic that we picked up with Rory Kethen-Jones as well. You know, the fact that social media, of course, has brought us many wonderful opportunities to learn and interact with people. But this topic of misinformation, and I was just curious. I mean, I know there's been this debate with Twitter and, you know, what Elon Musk might do it had he acquired it. We won't go there. You know, we've always said that, I mean, ultimately, science has to be the arbiter of the truth. I mean, as far as, you know, in the Popperian sense, you can get to the truth, but it has to be. So with that in mind, how could science play a role to in some way moderate social media in these areas without interfering with its essence?
Yeah. So the short answer is I have no idea. The long answer is I think so Twitter is making some efforts. I've given them some, you know, very often. So very unpleasant interactions, very aggressive interactions will be removed immediately. And the problem is that becomes a badge of honor to some of these people, and they will try to get themselves back.
Yeah.
You see this. Yeah, and so that's a. But also getting and Twitter does try to identify things that are untrue and dangerous, and we saw this during the pandemic where they were often signposting, they were showing things around, but that's very, very difficult to do at speed, and often you would see the same very loud-mouthed person saying many things, even if 75% of the time the same comment would pick up a flag from Twitter that it's that this is untrue or likely to be untrue. There'll be many places where that wouldn't happen and they would get shared. So how would I and how do we help people realize that they're dealing with a crank or a snake oil salesman versus a scientist? Because many people will claim to be scientists and aren't. And there is one very large Twitter account of somebody under the name of a scientist, but a scientist who sadly died some time ago. So it's quite clearly not that person, but yet they have very, very many followers. It's interesting that several scientists, several people I know, several friends have got that blue tick, and I think that helps. And that's usually because they've written articles for newspapers. It's very difficult for scientists or medics to get evidence that they are real accounts, because it isn't something currently that the social media consider valuable.
That's really interesting, because we were thinking about using online sort of, you know, whether it's ResearchGate or some online means to actually validate people with scientific or medical background. It's interesting. So yeah, I agree. As you said, it's a difficult topic, but, you know, people are out there talking today. So it's one, you know, that sort of rages on and has to be tackled in some way. I wanted just to move, we've been talking a little bit about your clinical work and your clinical research as well, although I think you do other things in clinical research. But can I move rather tentatively towards the topic of protein folding? Because it's going way over my head, and it may be for many people, but it's fun to understand if you can explain to ordinary human beings. I think I sort of know what a protein is, but not even sure about that. What is it that's going on there? And I actually did a quick search on PubMed, and you appear to have 101 citations and quite a lot in this area. It seems to be endoplasmic anyway, you tell me anyway.
Yeah. Oh, no. I'm more than happy. You're going to have to shut me up at some point. So the proteins. So this is go back to school biology now. So to remember, the cell is basically a complex bag of chemical reactions. The outside is lipid, so basically a thin layer of fat, and inside it's a solution of bits and bobs. And there are the structural bits, which are generally proteins. Things, like meat is all protein mainly, but every cell in your body has structural bits and mechanical bits which are made of protein. And then there are things like stores of energy, like carbohydrates, sugars, starches. And then there's a lot of the information is stored as a different type of molecule called nucleic acids, so DNA or RNA. And that's pretty much most of the things. So the proteins are the mechanical bits. They have to have a particular shape, but everything, they're made by joining little individual components together called amino acids into long chains of hundreds and hundreds of these different amino acids. Now, a long chain is completely useless, but it folds like some incredible biochemical origami into this three-dimensional structure. There was when the same time there was the Rubik's Cube, there was also that Rubik's Snake. Do you remember the silly thing?
Oh yeah, I remember that. Yeah, I couldn't do that either.
This is like, imagine that shape. That's the protein, and when it's all out and you can't do it, that's when it's first been made. So it has to fold, and you can fold it into different shapes. You could get the globe, or you can get all these funny things. That is how proteins work. But those individual components, those amino acids, make it want to fold in a particular way. And that's why the protein that makes up hemoglobin, the thing that carries oxygen around your body, has one shape. The protein for a receptor that responds to a chemical in your brain to make you think this or the other has a different shape. And that's determined by the order of those little amino acids. So some proteins, simple proteins, if you had them in a simple string and you put them in solution in some liquid, they would eventually fold themselves into the right shape. But many things are much more complicated than that, and there has to be a complex machinery inside the cell that as the protein is being made, it's folded into the right shape, and then it can go off and its job. But very often that's so complicated that a large minority of proteins that are made never get to their correct shape. So they get folded into some wrong shape conformation, which is unsalvageable and never can be used. And so they have to be recognized and disposed of, and the cell starts again. If lots of those bad versions build up, that's called proteotoxic stress, right? Proteotoxicity. And where it happens in the cell has different varieties. If that's not niche enough, I'm specifically interested in that happening in a part of the cell called the endoplasmic reticulum, or ER.
The ER, yeah.
So ER stress has got nothing to do with the emergency room. It's the part of the cell which makes proteins that are going to be secreted, like insulin and things.
And what happens then if you have a problem in that area? Or
so if loads of them build up and they overwhelm the endoplasmic reticulum, it stops working and the cells die. And that's actually, turns out, it underlies a lot of human diseases. So there are diseases with ER stress affecting the brain. There are a whole host of them. ER stress probably plays a role in lots of forms of neurodegeneration. It plays a role in lots of forms of diabetes, and I can go on. It also happens, and so the body, evolution has provided us with lots of mechanisms to detect it and try to defend against it and spot when it's going wrong and fix the problem, and that's something we study. Now, when it's going wrong, you usually want to fix it. But if it's going wrong in a cell you don't like a cancer cell, you would want the protective mechanisms to stop working. So because of research in this field over the last sort of 20 years or so, we now understand the defensive mechanisms so well, we can interfere with it. It's easier to make things not work than it is to make them work better. So currently, the drugs that are aimed at the ER stress type field are actually to make it worse. So there are some anti-cancer drugs already in use, and there are lots in development which are designed to make our own response to ER stress.
That would be quote unquote immunotherapy of some description.
So no, immunotherapy is slightly different. Immunotherapy is outside the cell, and it's where the cells are talking to each other. So cancers are often destroyed by the immune system all the time, every day. Scarily, it's probably happening to us right now, but we're not getting cancer because they're being fought off. But cancers that are successful from a cancer's point of view are ones that are good at turning off the immune system locally. So immunotherapies are designed at turning up the immune system locally. Now, in a similar vein, inside the cell.
So this is inside the cell, turning it off. Okay.
Inside the cell, cells that are cancer cells often have a lot of ER stress. They're making loads of proteins, and often they're making mistaken ones. So the cancer cells, many cancer cells, can become essentially addicted to the defensive mechanisms that normally protect us. So a lot of the new advances in my field are to try to just slightly weaken our responses to ER stress. So most of our cells will cope because they're not suffering that much stress, but the cancer cells, which are really addicted to these defensive mechanisms, will then fall apart. And we know in things like multiple myeloma, which is a cancer of cells that make antibodies, incidentally, forms lumps inside bones. Some of the drugs that probably now are the most effective are the ones that make ER stress worse. Okay. And it's quite remarkable that patients who would have been refractory to any treatment 20 years ago can now be put into remission, and that's with the first generation of these drugs. And there are many, many more coming through that are in sort of the preclinical, pre-patient stage, which are very specific. And the hope is that we'll actually have a whole new suite of anti-cancer drugs.
Okay, wow, that's amazing. Go ahead, Laura.
Sorry, Andy, I was just going to say, well, it's always the sign of somebody brilliant that they can explain something so perfectly that it makes you feel that you understand it, even though it's well beyond your sphere of understanding. But what did strike me listening to you and knowing how sort of fascinating it is for both of us is the real change that I think has taken place in healthcare over the sort of last generation, where people's relationship with their doctor and with medicine in general was quite sort of disparate. And I think patients would go and see a clinician and they would be told what was happening. And we have that sort of culture within the NHS where doctors communicate with each other and patients are copied in. But I think one of the things that sort of shifted, and slightly with the wellness industry, is that people are being encouraged to be more engaged in their own health journey and to take some more responsibility where they can of promoting wellness, and particularly sort of in the field of preventative medicine. I just wondered, from your perspective, how important it is for people to really develop their own understanding of sort of body systems. I mean, obviously in a way that's kind of intellectually appropriate. And to be engaged with that process and journey of kind of wellness.
Yeah, I do think it's important that people to understand. It is unfair if we expect patients to become experts in the same way that I'm not expert in cardiology and expert in neurology. Expecting every patient to become completely expert in all aspects of medicine is difficult, which is why signposting people to appropriate advice. And that's why, the snake oil salespeople who have a wonder type of diet are able to make so much money, because they're able to con people, they're able to feed people some really nice, simple untruths. The truth of the matter is biology and medicine is incompletely understood, and that can be sometimes quite uncomfortable. So it's not that I don't think patients should be helped, assisted. I just think we have to be realistic. We also have to we have to help people by starting to teach people very early on in life about uncertainty, and how when two specialists disagree with each other, it's not necessarily the case that either of them are wrong. And that's one of the problems we saw during the pandemic, when people had slightly different opinions, that meant, they're both wrong. You know, there is a debate and therefore it's not understood. But that's biology, science in general, but certainly biology at school is taught as a very nice, simple round that you can know everything and pass the exam. And if you say this phrase, you get this mark. In reality, when students come to university, they get a real shock and they find out that the real world is not like that. And that the world is so incredibly complex, it doesn't always fit into a simple, understandable method. So it's important that patients understand their disorders as well as they possibly can, but also appreciate that there are gaps in our knowledge, and that disagreement between specialists does not mean an absence of truth. Is that making some sort of sense? And that's why we need to signpost people to reasonable opinions.
Yeah, and by the way, I think that's also something that we've debated a lot about, trying to help people, signpost people, to help them navigate through the world of health and wellness, and ultimately get to the right people to help them move forward with whatever topic they have.
I think there's also the element, isn't there, that medical care, you know, in terms of like human history, is such a new discipline.
Yeah.
You know, it's only sort of within, you know, a few decades that we've actually had the first heart transplant or something like that. And so I think it really highlights as well the importance of recognizing that the clinical practice is really sort of still emerging in so many ways, and the importance of collecting experience and data to really to move that forward.
And changing our minds when we learn more. Exactly. So, and again, when I'm teaching medical students, and they're sometimes a little bit perplexed that the first 2 years is essentially doing natural sciences, essentially doing basic sciences, certainly the way we teach it at Cambridge. And they're thinking, well, why aren't I on the ward seeing patients? Why aren't I taking blood pressures? Why aren't I, doing examinations? The thing is, if we teach people the understanding, the basic science, we future-proof them. The medicine that I'm practicing now is very different to the medicine I would have been taught when I qualified 25 years ago. We would never have given beta blockers, the drugs that slow down the heart. We'd never have given beta blockers to somebody with heart failure. It's just, we used to stop patients who had beta blocker eye drops for their glaucoma if they came in with heart failure. We now know, because of research that's been done subsequently, there are many situations where we want to give beta blockers to people with heart failures because it increases their likelihood of surviving, and because of various complex interactions. But it's an understanding of the basic science and a realization that when the science changes, we change our minds. That's not a bad thing. That's a good thing. And also, as you said, the technology. It cost 5 billion and 5 years to do the first genome about 20 years ago. Now, when I see a patient in clinic, several times a month, I'll be saying, let's just do a genome. And so we do a genome, as this is already current medicine. I mean, we don't look at all 20,000 genes. We'll do the genome because it's as cheap to do a genome and then look at the 20 genes I'm interested in than it is to do 20 individuals. But certainly over the next 5 years, and certainly by the time the students I'm teaching now are consultants, whole genome sequencing will be an absolute standard of care.
On the topic of genomics, because I really wanted to get this question in, which is because we've obviously got people who are quite interested in tech in the broader sense, the consumer sense as well, and of course, when I saw that you were involved in protein folding research, I thought immediately about AlphaFold.
Yes.
And I think CASP, I don't know if CASP has taken place this year. It must be CASP 15 this year. But anyway, the idea, at least for as far, I tried to watch a video and saw how much of it I understood, which was also challenging. But I think basically taking a genome sequence and then predicting how the protein would fall. But I think this whole area of applying particularly deep learning advances to this topic, I'm just curious how much it affects your work and what you see coming out of all the collaboration of the two disciplines.
No, AlphaFold. So for those people who haven't come across AlphaFold, I mean, maybe your listeners from all know it. AlphaFold is this amazing, really, really shocking new, basically, software that came out within the last 12 months, which, by looking at the shapes, the structures of thousands of proteins and their sequences, and applying some very clever computations that I don't understand, it's able to make the most extraordinarily good predictions from, if you give it a sequence of an unknown protein, it can now very often, not perfectly, but very often predict what its structure is like. So much so that most people now will quickly go to AlphaFold, and if AlphaFold has a reliable structure, they'll assume that's right. Now, that won't always be true, but it's very, very good. So many, so crystallizing, it's still important to crystallize proteins and work out their structure and use new technology like cryo-EM, but to make sure these structures are true. But right now, something like AlphaFold is a huge step forwards. What AlphaFold is not very good at is predicting precisely what a mutation will change about the folding. I am sure in 5 or 10 years' time, we'll have wonderful computational methods of linking a new mutation that we find in one of our patients, yes, to the structure of the protein and maybe the effects of the protein. But right now, we can't really do that. So we very often now, we do genome sequencing or whatever on patients. We find, if I were to genome sequence you, so you were to come to my clinic with a pneumothorax, and for various reasons, we decided, it's not always the case, but we do a whole genome sequencing of you, we are likely to find many genes with apparently pathological defects in them, because we've all got them, but most of them don't matter. Because we've got 2 copies of things or various things, we can compensate it when we develop. So to say, well, actually, you've got that condition and you've also got these several dozen defects. How do I link which one of them or several of them are linked to your disease? Well, the first thing we currently have to do, and people in my lab have to do, and people in labs around the world have to do, is put that mutation into the protein and then study it in cells or study it in the lab. Right. And sometimes the software does make the right prediction. It says this, if you've got a mutation that cuts the protein in half, because the second half of it can't be made, that's probably going to not work. But if it's got one change of the letter, so it doesn't fold that way, it folds that way, yeah, it may still work. The protein may still work at its job. So you currently have to do the experiment. And so Julia, one of my postdocs, for example, is spending a lot of time doing computational stuff to try and make predictions. But in the end, she's still got to go and study the protein, and it takes months to do it well. And that's something we're going to have a lot to deal with over the next 5 or 10 years as it becomes more and more easy to get the genetic information. How we turn that genetic information into knowledge about a disease actually takes a lot of lab work.
And so are you working closely with the computing department then in these areas?
So I'm not doing this computing department. So for example, I've got a new PhD student starting this autumn, and hers is to study mutations in a particular protein that's defective in a particular lung disease. And certainly, Julia, the postdoc's working on that project is looking at various known defects or changes in that protein that have been found in patients that we are not yet sure whether they're the cause of it. We know that they're present. We know the patient have got that disease. We don't know if the two are linked. So this new PhD student will be working with me, but will also be working with a structural biologist in my department. So Janet Dean, one of the structural biologists, her job, rather than looking at how the cells respond to ER stress like my lab does, hers is to actually solve the structure. So we talk about when you've got a protein that a new protein, and you figure out what it actually looks like. That's called solving the structure, because you often will make crystals of it, shine X-rays through them. They'll be scattered in incredibly complicated ways, and then crystallographers can do extraordinary computational magic and work back, see what the protein shape is like, and that's what Janet's involved with. So that's where you've got a cell biologist like me working with another field, a structural biologist, and together the interface is much more. So I wouldn't pretend to be a structural biologist, but I know a lot of people who can do it, and then I just work with them.
So it's obviously exciting the way that's sort of propelling research. And I think in the case of, I think even at COVID in the early stages, the genome was made available quite early on of SARS-CoV-2, and
it was the first half of January. So it was an extraordinary time for many years. It was an extraordinarily bad time, and I wish we hadn't had to go through it. But it was, the disease was first appreciated at the end of 19, beginning of 20.
Yes.
The Chinese, some extraordinarily brave Chinese scientists and doctors were looking after these patients. They sequenced the genome and made it available. I have to check the date, but it's something like the weekend of the 10th or the 11th of January, something like that. Rather than waiting to publish it as a research paper, they simply made it available around the world immediately, which was a game changer. Because of that, because that genome became available like that, researchers around the world that were making new vaccines, for example, these new mRNA vaccines, which are extraordinary because you can change them very, very quickly, the whole infrastructure was developed to make RNA vaccines against another virus. But because the sequence of SARS-CoV-2 became available at the beginning of January, many scientists around the world were able to pivot towards this new threat. And that's the only reason why by the spring people were able to do preclinical studies. By the summer, clinical studies. The data were being fed to the regulatory authorities such that we had actually licensed medications by the end of the year. If you had asked me if that were possible back in 2019, 2018, I would have said no, that's completely unrealistic. But it was this sharing of data. And being able to sequence a virus like that, and then sharing it around the world, that meant that we're now sort of beginning to head towards something like normality. We're not there yet, but we're closer to normality because of that data sharing.
So as traumatic as the whole period has been, it could have been a lot worse.
Oh yes, I mean the death toll is horrific. But it would be much worse. It would have been much worse had we not had virus, had we not had effective vaccines. If we hadn't had large, there were, in addition to some vaccines. I mean, vaccines have really changed the game in COVID. But the studies, the new type of research studies, the relatively new research, rather than just doing one drug against nothing or one drug against the old drug, the old-fashioned way. We saw this platform trials. This is where you have many drugs being tried simultaneously, and constantly the losers being dropped out and being replaced with the new one, and evolving over the weeks in front of your eyes. Yes, those studies theoretically existed beforehand, but they've now come into their own, and they came into their own because of the pandemic. And I think it's not a coincidence that many of the big advances in COVID drug treatment, and certainly the first one, the first effective treatment was dexamethasone, came from the UK. And that's because we have a joined up National Health Service where all the doctors are working together for the benefit of everybody rather than for profit, teamed up with the NIHR, the National Institute for Health Research. Basically, these work hand in glove, and they were able to develop these platform trials. So basic science, sequencing, new trial methodology has reduced death toll massively.
And do you think it's sort of enacted any kind of cultural shift within the scientific and research community, having that initial sharing of information slightly against precedent that is going to continue? And just as well, sort of interested in your thoughts about the responsibility that first world countries have to the developing world in this area as well.
Yes, so two really important questions there. So the existing scientific model, I'm not saying is completely broken. I think the peer review system. So for those people who aren't familiar, if we do some research, I can believe all the things I've done, and I think they're great. Write the paper. It's sent out to a journal, and a journal in an anonymized way to people I don't know who owe me no favors and will take great delight in finding all the faults in my papers, will criticize the bejesus out of it, then send it back. The journal will either say this is garbage and reject it, or saying, well, look, Marciniak, have it back. These are the comments, try and fix them. And then after several rounds of that, you end up with a much better paper, and a paper which the community will benefit because it's had a huge amount of sense checking. That's a great system. It's slow. And so I'm not saying we should throw it out completely, because we can't lose the baby with the bathwater. But there is also a lot to be gained by early sharing of information. And it's now becoming quite normal for people to write their paper, and then make it available to everybody on a sharing platform, something like medRxiv or bioRxives. These come with a huge health warning that these are unpeer-reviewed papers, right? And a lot of people, people who, particularly people who aren't in the scientific community, will not always distinguish between them. And so there is some garbage gets onto these bioRxives that would never get published. And they are certainly not as valuable as a high-quality paper, but at least they're out there to be discussed. People need more tools in how to understand science to be able to separate those ones out. But it does mean, if I do a paper and I can share it, that I can benefit from other people's views about that paper when it's on bioRxives, and improve it even before I submit it to a journal. Or I can put it on bioRxives and let several journals look at it simultaneously, and the one that likes it the most pick it and send it out to official reviews. So I think this early sharing of information this bioRxives was taking off, 2 or 3 years before the pandemic, but it's really become normal now. The physics community were doing it for years. The physics community were way ahead of us. They were doing this for many, many years. The biologists and medics were only doing it more recently. So yes, it's sharing early, sharing widely, but giving people the tools to spot when something is peer-reviewed and when something isn't. So yeah, that's sharing information. Sharing large data sets. It is completely wrong if people do large pieces of data not to share the information, and most journals won't accept it. If I do some research and I do lots of genome sequencing, or I do lots of RNA sequencing, all of that information might not go in the paper, but it has to be accessible to the community. It has to be put on national, global archives in an anonymized way that other people can mine those data. There's experiments I did back when I was in the States in 2014, where the data is still available and people can still access it because there's far more data generated that I could use. So yes, so I think sharing early is really important. And the second question I've already forgotten. It
was about the responsibility of first world countries to kind of share and improve standards.
Yeah, so there's several things there. One is sharing research, and I think it's very, very normal in the UK with any of the big funders, any of the government-related funders like the MRC or the EPSLC, or the other big funder, the big charity funders like the Wellcome Trust. It is completely unacceptable to publish research which is not open access. And that means that anybody on the planet, whether they're your mate down the pub or somebody in the developing country, can just go to it, click on it, and read it without having to pay $35 to read it. So journals being able to guard all of our research that we've done, and they guard it behind their firewalls and insist that everybody, including people who can't afford, have to pay to access it, is gone. So I can't even, my university can't count any of my research papers towards the 5 yearly annual reviews of all universities in the UK. They can't count one of my papers as being an output of the university unless it's open access. And I think that is a great thing. So that's one thing to help researchers in those countries. The other is to study diseases that are relevant there and aren't simply relevant here. Obviously, I think it's important to study rare genetic disorders that affect a small number of people in the UK, and I'll continue to do that. But I absolutely fully support a huge amount of effort going into diseases such as TB, malaria, which kill far more people in far less advantaged countries. We research, one of the aspects of the research in my lab is a particular cancer called mesothelioma. It's a cancer caused by previous exposure to asbestos. And I once had somebody from an insurance company say to me, "Why are you studying?" I'm not kidding. This is a critical. They said, "Why are you studying this disease? Because it's going to die out in the UK. We banned asbestos a few years ago. Those patients with it, over the next 10 or 20 years, it's going to become less of a problem in the UK because fewer people will have it." First of all, that was a shocking thing. So I can't believe that they suggested that. But secondly, there is as much asbestos being used on the planet now as was used in the 1960s in place when you just considered the US and the UK. And that's because countries like Brazil, Russia, India, China are using asbestos now because it's very useful in building materials. It just tends to kill you 30 years later. So the research that we're doing on that cancer here will help developing countries in 20, 30 years time.
Yeah, and I think it's also something that we feel strongly about, and also it came up in the interview with Rory as well about the fact that also not only could information be shared from perhaps a developed economy to a less developed economy, but there may well be the answers may also be around in another country that could benefit the UK as well. So it could be a two-way street.
Absolutely. So researchers in South Africa are absolute stars when it comes to sequencing. You know, so the BA2, the BA3, the new variants that were analysed and studied. These are sort of offshoots of Omicron that were analysed and identified in South Africa before they even reached other countries. Now, we don't know for a fact that they even started in South Africa. We just know they were first detected there. And actually, there's something to be said that those countries were penalised for identifying them. But lots of extraordinarily successful science is done in developing countries, and they needed to be supported. They need to be supported to do that, not by paternalistic scientists going over there and telling them what to do, but working with them so they can co-create a scientific infrastructure within their own countries. And we all benefit. You know, we all benefit if we're not simply doing all the sequencing in developed countries, that if it's being done in places like South Africa.
And that brings me, maybe I know we're running for. I feel like we could go on talking for hours. This is fascinating. I have one question. I know, Laura, you may have others as well. Just sort of connected to that, the topic that we're also interested in, which is how can people, patients, caregivers share data in a responsible, controlled, de-identified way with researchers for good purposes, and I think, again, from the interview with Rory, it came up. We have this great awareness of data privacy, and that's good in many contexts. But in the context of sharing it for research, we actually want to encourage people to share their data now, and they have increasingly, have wearables as well. So yeah, I'm just interested how you see that topic.
So I completely understand people being anxious about their medical information. There are many examples in the past where people's information was used against them. To people's back in the day when HIV was a disorder which killed patients very, very quickly. Having an HIV test, even admitting that you'd had an HIV test, had the potential to change your insurability. Of course, it's not relevant anymore, but back then it was. So people are understandably sensitive. And so when efforts were being made to share clinical data at scale, say from GP computer systems or from hospital computer systems, a lot of people opted out. And I completely sympathize with them doing it because, there have been data breaches in the past for other things, and we know. But I think it's a hugely altruistic thing to do. I'm very happy for my clinical data to be shared and used in an anonymized way, because it's when you aggregate data from thousands, hundreds of thousands, millions of people that you can see patterns that aren't available when you're looking at small numbers. So I think trying to persuade people, trying to make them understand that sharing these data with the research community, but also, and this sounds a little bit distasteful, companies, is necessarily a good thing. I don't get money from companies that use these data, but they have a massive, they have a huge number of medications that they need to work out which to prioritize, and we will benefit if they're turned into drugs. And they also have huge amounts of resource and computer expertise, and they can mine these data. So I know people find it distasteful to involve companies, but it is a model that works, and not everything can be done by small research. I've got 10, 12 people in my research group. There's no way I can manage the sort of scale that large international companies can do. We need governments that we can have trust with, that we know that they will make decisions for our benefit and that we can support them. And that's why trust in government is such an extraordinarily important thing and very easily lost. Patients, though, in general, I find, are very altruistic. Yeah. Mesothelioma, as I said, most of the patients who are diagnosed with mesothelioma will not do well, but they're very keen to be in clinical trials in the full knowledge that they still won't do well, because they want people who are diagnosed with this condition in 10 years' time to have treatments. You know, when we set up mesothelioma tissue bank, so we can supply researchers with tissue from this rare cancer to study, once it became available, we were contacted from people around the country saying, I've got mesothelioma, when I die, can I send you my body? And it doesn't quite work like that, but people want to do that. Yeah, so we need people to understand the benefits of sharing information and being involved in clinical trials, the benefit for the community, and we need them to trust. And the way we trust with them is explain to them what's going on. So the sort of work that you're doing is really, really important, and we need leaders that we can trust.
I think it's such an interesting point, and always with these conversations, there's so many parts that are kind of adjacent to each other. One of the other key aspects that's kind of led to the development of Care Circle is thinking not just about that kind of quantitative data that's so important, but also the sharing of people's experiences. And you touched there on sort of trusting governments, you know, the kind of cultural issues that surround medicine and treatment. And you mentioned HIV, which obviously was such a political topic at one point and still is for many places around the world, but also thinking about what's happening in the US around Planned Parenthood and that topic, FGM, just really thinking about the importance of patients and people sharing their experience to try and really sort of like democratize the information that's available to people, perhaps where governments take a view that isn't perhaps in line with what the medical community might consider to be good practice or in the interests of the patients. And just wondered really what your thoughts were about that.
So one thing that's now normal in medical research. So I have my medical research hat on and my laboratory research. When I do laboratory research, I write science grant applications that probably only a small number of scientists can understand. They're so incredibly niche. That is not acceptable in medical research. In medical research, there's always an abstract, a summary for the scientist, but there's also a lay summary. And you know, and if your grandma can't understand it, that's not going to fly. It's very important these days whenever people are applying for medical research funding, it's the grant bodies, and I'm on various grant committees as well, and we absolutely expect that any grant has been put together, any application and project has been put together with the involvement of patients. And this can be done at many levels. A very, very high level, there are, every so often, there are, for various diseases and various conditions, there are priority setting exercises. So there's an organization called the James Lind Alliance, and it helps large communities of doctors, patients, charities get together and over the period of months and exercise, sort of a conversation really, develop the priorities. So what both the research community, but as important, what the patients think are important. Because what I think is important to do with research may not necessarily be what a patient thinks is in research. Now, I think curing mesothelioma and making somebody live some months longer is more important. The patient might think, well, actually, you know, I'm okay with living a year. I realize that, but I want to spend less time in hospital. So involving patients in the research and priority setting is one thing. So that's the very high level. At the next level down is in terms of developing a research project, involving patients, asking them if this sounds okay to them. Are these, you know, if there are questionnaires involved, if there are consent forms, having patients see those before you actually finish the grant is really important, because patients will look at that, sometimes look at the consent forms and say, nobody's going to understand this first page, or you know, this seems a little bit off. You can't ask the question in that way. And you know, we're all humans, we should think about it, but we've not lived the experience of having that disease and been to the clinics and been terrified. And so involving patients in the development of research is absolutely required. In many grant applications these days will often have a patient as a co-applicant. This is quite normal.
Well, I think that's a great place to finish the interview. Trust patient interaction with researchers and medical professionals. That's what we aspire to support. And Professor Marciniak, Stefan, thank you for your time today. It was fascinating, and yeah, we hope to be in contact, and thank you very much for your time.
I'll be chatting with you on Twitter, no doubt. See you.